nice to be with you tonight. And before I begin, I just want to thank the chapel committee for inviting me to speak here with such a wonderful array of scientific professors. I'm a mere physician, kind of a petunia in an onion patch, or vice versa. I know you enjoyed Bob Latimer last week. I've known Bob for many years. Bob is a friend of mine, but I am no Bob Latimer. I'm not going to be quite as erudite as Bob was last week, but I hope that I can maintain your interest. Having said that, I want to make it perfectly clear that I firmly believe in the concept of intelligent design. But I am of a particular subspecies of that species of scientist. For example, in the area of theology, I'm a theist. There are many types of theists, but I am a subspecies of theist known as an evangelical Christian. And my theos, my God, is the triune Father, Son, and Holy Spirit God of the Bible. Now, my subspecies in intelligent design is that of a biblical creationist, and I make no apology for that whatsoever. My belief in intelligent design based on creationism is because of the collective truth that I observe in both natural and special revelation. Natural revelation being the world around us, and special revelation being the Word of God. As C.S. Lewis once said, I believe in Christianity and the Bible for the same reason I believe that the sun just came up. Not because I see it, but because by it I see everything else. That's my perspective on intelligent design and creationism. Now, the whole enterprise of science is predicated on the notion of seeking the truth. Truth is absolute. It's not relative. Truth is not a product of the cult of materialism. Truth explains the existence and purpose of materials. And truth transcends naturalistic theory. Now, you should not want to be lied to by a biologist, a physicist, or a chemist any more than you want to be lied to by your personal physician. Think about that. And yet, in the arena of origins, we have seen a paradigm shift in the past century. Truth has been suppressed in exchange for an illogical lie. An illogical lie. As a result, the 20th century has been declared to be the bloodiest in all recorded history. And the 21st century has not gotten off to much better a start. Now, unlike evolution or revolution, the word devolution is not a household term. As a matter of fact, you may not have even heard of devolution before. Let me give you the definition of devolution. This is from the dictionary. It is the transfer or inheritance of authority from a monarch to his or her successors. Now you might say, what in the world does that have to do with what we're talking about tonight? Well, here's what it has to do with. Over many years of being educated in the theory of evolution as preparation for becoming a physician. The theory of evolution had gained intellectual authority in my mind and had clearly ruled whenever the subject or question of origins was raised. It was king. There was no other authority. However, in June of 1970, that authoritative, fallacious doctrine was overruled when I had a life-changing encounter with the living God, the sovereign, omnipotent, omniscient creator of the universe. And as a result of that transformation, my worldview was dramatically changed toward a variety of issues, not the least of which was that of the origin of the universe and biogenesis. Biogenesis being the origin of life on planet Earth, the only place where life has ever been found. Truth took over as monarch in my mind, thus the devolution of the fallacy. Now, for years as a result of my undergraduate and medical education, I had been awash in the Kool-Aid of evolutionary thinking. Or maybe I should say, in order to not mix my metaphors, the Kool-Aid of evolutionary drinking. I had actually not been thinking for myself. I had been allowing my professors to think for me. And there was only one way to think if you wanted to get the questions right on the exam. Thus, in 1970, when I began to ask myself where the science was in this so-called theory of evolution, I reasoned that it didn't really exist. It was not in any way consistent with the scientific method that I had been trained to obey throughout all my years of study. The virtual sine qua non of all true science is the scientific method. Now, as all scientists know, that anciently derived method begins with an observation of certain facts. Having observed the facts, the question then arises from the inquisitive observer, why this instead of that? How did this happen? Why did this happen? What could have made that happen? Why didn't something else happen? And so the reasonably thinking mind will posit a possible answer in the form of a hypothesis. Now, it's perfectly all right to theorize. As a matter of fact, the late Linus Pauling once said, the best way to have a good idea is to have a lot of ideas. And he did. Well, after positing a hypothesis, the true scientist then establishes a laboratory experiment to prove or disprove the theory. Next comes the result of the experiment. And having, on the one hand, disproven the putative hypothetical answer to the why or how question, then one abandons the erroneous solution and conceives another explanation, another hypothesis, for which one sets up another experiment. On the other hand, if one's hypothesis is validated by the experiment, the testing is repeated. And if the same result occurs, one has proven the hypothesis. The idea thus moves from the theoretical realm into the realm of factual things, and the findings are reported in a scientific journal or forum. Now, in regard to the theory of the origin of the universe and first cause, I had been taught about the Big Bang. I had been taught that it was factual and that it had been a thermonuclear explosion of matter and energy that occurred in a matter of nanoseconds, and that the result of that is continuing as the universe expands. And at last measure, it had expanded to a point of 15 billion light years beyond planet Earth. And as I pictured that in my mind, I said, expanding into what? You ever think about that? What's it expanding into? And what's beyond the expansion? Well, nobody's figured that out. But supposedly, that thermonuclear explosion 15 billion years ago had caused all the design, order, purpose, and life that we see throughout the universe and especially on planet Earth. Think about that. A thermonuclear explosion had caused order, design, purpose, and life. Good hypothesis. On the other hand, it has been tested in the laboratory. Now, applying the scientific method to such a theory, I realized, first of all, that no one had been there to observe the so-called Big Bang or ask why or how it happened. It's kind of like back in the days of the Cold War, one guy was sitting on a fence and another guy came by and he said, do you know what you should do when the bomb goes off? And the other guy said, it don't make no difference as long as you can say, what was that? Well, there wasn't anybody there to ask what was that 15 billion years ago. So we've missed the first part of the scientific method. Then I remembered that another thermonuclear explosion of a much lesser proportion than that proposed in the Big Bang had already been tested twice. Once on August 6, 1945, in Hiroshima, Japan. And again on August 9th, 1945, in Nagasaki, Japan. I was only 9 years old, but I remember it well. Needless to say, those Big Bangs had not resulted in design, order, or life. Observable facts had determined the exact opposite. The hypothesis of the Big Bang as the first cause of the universe remains utterly and totally invalid, and it should be discarded, especially from the classroom, based upon the scientific method. Next, it ultimately came to my newly reasoning mind that the Darwinians had also proposed a first cause in biogenesis. Now, I'm not a nuclear physicist, but I am a physician, and biogenesis and life is what I have given my life for. And I am a firm respecter of the sanctity of life. And so, their theory proposes that a single living cell, a self-sustaining, self-replicating, highly complex creature had come into existence spontaneously by the action of some inexplicable natural phenomenon, suddenly impacting a theoretical admixture of organic and inorganic substances suspended in a primordial sea. That's the hypothesis. Okay? Okay. That's okay to have a hypothesis. In other words, life had come from non-life through impersonal nature. Now that totally disregards the well-respected cell theory that all biologists look to. And the cell theory has 3 axioms. The first being that all living organisms are composed of cells. There are creatures that are unicellular and creatures that are multicellular, but they're all composed of cells. The first creature allegedly being unicellular. The second axiom is that the cell is the basic unit of all life, animal, vegetable, and human. And the third axiom is that all cells arise from pre-existing living cells. Okay, again, I applied the scientific method, and as to step 1 in this theory, observation, no one had been there to watch or question that alleged biogenic event on Earth that reportedly occurred 3.5 billion years ago. No one had been there. And so, a hypothesis, step 2, had been formed apart from step 1, thus ignoring the scientific method once again. Now, as to the experimental phase of the method, step 3, we have on multiple occasions observed the effects of raw nature on our surroundings. Think about it. Lightning, earthquakes, hurricanes, tornadoes, floods, and volcanoes do not appear to bring life from non-living things. It's never been observed. What is science? It's observation. It's okay to have theories, but discard your theory once you don't test it and find that you've proven your theory. And time again, we've had tornadoes and earthquakes and hurricanes and slippery roads that cause 18-wheelers to spin out of control and kill people, but no life has come from non-living things. As a matter of fact, spontaneous generation was disproven by Pasteur hundreds of years ago. Someone had been walking along the road one day and had seen a cow laying in the field. He came by the next day and the cow seemed to be riding. He thought, ooh, cow's coming alive. But the cow didn't smell too good. He came by the next day and there were these little white things crawling all over the hide of the cow. And he thought, wow, life sprung from non-life. And then, of course, Pasteur went and proved that you can kill the fly's eggs long before they hatch into maggots. And field laboratory tests, so to speak, have failed to uphold the hypothesis of some natural phenomenon impacting anything and bringing about life. Now, furthermore, having taken the scientific method to another, more deliberate level of experimental stage, evolutionists have never, in 150 years of trying, ever produced anything close to life in the laboratory, despite their superior intelligence and the intricate designs of their experiment. Now, what if they perchance do bring life out of their intelligence and design? Well, they have proven the concept of intelligent design. It didn't happen by happenstance when some lightning bolt hit a frog pond. Finally, and we're going to get finished with the discussion here in a minute. Finally, and we're going to get into science. Having been involved in the art and science of medicine since 1957, now this is very important, I have never observed nor read of a mutation that ever resulted in an improvement in a human's condition. Think about that. The whole Darwinian theory is based upon random mutations and survival of the fittest by selecting out those most fit to survive. In the human experience, those who have mutations that cause cardiac problems or pan-body problems like Down syndrome or muscular dystrophy or hemophilia, they don't improve one's chance of survival. They diminish it. And so, once again, in the field, the test of the hypothesis has failed to prove the theory. Now, having reasonably and logically discarded the notion that we humans are the byproduct of time plus matter plus energy plus chance, I have been able to see, as a physician, the wonderful design and purpose in every aspect of the human body, in its gross and cellular anatomy, in its biochemistry, and its physiology, for they all speak quite loudly of what the Greeks call telos, or purpose. Everything in our body has a purpose. You know, when I was early on in my medical education, there was a lot of talk about vestigial organs. Now, a vestigial organ, first of all, there are no vestigial organs in humans. Everything that was called a vestige of some previous evolutionary change in the species proved to be not a vestige, but a functional organ. There was a time when the thymus was thought to be a vestige. Now, it was called the thymus because it was thought to contain the soul. Interesting. As a matter of fact, another name for depression is dysthymia, okay? Which is a disturbance of the soul, or the psyche. And so the thymus was named to be the housing of the soul by anatomists. Well, we now know that the thymus is one of the most important immunological organs in the body. As a matter of fact, the AIDS virus attacks the thymus and interferes with several of the thymocytes that do several things to prevent cancer, to seek out cells that are degenerating into malignant forms and taking them out. They also help, they're called helper T cells, and they go around and help the B cells, which come from the bone marrow, to fight infections, either viral or bacterial or fungal. So the thymus is not a vestige. It's a very important organ. There's another organ in the center of the brain called the pineal gland. Now, when I was a medical student, nobody knew what the pineal gland did. It has turned out to be very important in reproductive physiology, and also in the production of normal sleep, because the pineal produces melatonin, which is important in the sleep cycle. So again, vestigial organs are non-existent, and the notion that we humans are simply remnants of our evolutionary past is pure nonsense. Now, there is a notion that continues to appear in biology texts in the lower grades, not in graduate school, but in the lower grades, and it's based upon the fact that we theoretically evolved from single cells through various stages of animal life, fish, amphibians, and so forth, okay? And the notion is entitled Ontogeny Recapitulates Phylogeny. Now let me explain that. Ontogenesis is the process of becoming, okay? For example, the ontologic proof for the existence of God has to do with the being of God, okay? Well, ontogenesis has to do with becoming. So the ontology of becoming a human went through the various phyla, or various types of animals. So ontogeny recapitulates phylogeny. So what they did is they took the human embryo and they said, well, it all began as a single cell. Well, that's true. Yep, that first cell all began as a single cell, and then multiple cells, and then they showed the embryo with little slits here that they called gills, and showed the embryo with a nice tail like a salamander has. And these pictures appeared in medical books when I was in medical school. They still are appearing in some biology texts in lower grades. And it's propagating a lie. Now, I don't know how many of you have read Michael Behe's Black Box, Darwin's Black Box. If you haven't read it, I highly recommend it to you. And Dr. Behe is a biochemist at Lehigh University. He coined the term irreducible complexity as he studied the biochemistry of living cells. Basically, what he was saying is that in the biochemistry of life, for example, energy production that occurs in the mitochondrium, which is the powerhouse of the single cell, in that process, there are many steps in the chemical nature of energy production. And if one of those steps is missing, no energy occurs. Okay? And he used the example of a mousetrap. And he said that if the spring isn't there, the mousetrap won't work. If the lever isn't there to lock down the cheese, the mousetrap doesn't work. and so forth and so on. Any part of the mousetrap that's missing, the mousetrap doesn't work. Well, you know, complex cell is much more intricate than a mousetrap, but he used that to point out the ridiculous idea that anything could be missing in a cell, and that would all have had to evolve spontaneously, simultaneously, because if it didn't all evolve, that first cell could never have worked. Okay, well, he coined the term irreducible complexity. And being an independent sort, as I am, I decided to coin my own term for the anatomical reliability that the parts of the body have on each other, and I call it mutual dependence. Okay? Now, the microanatomical workings of the cell are mutually dependent, for example, in the production of a protein like insulin, or any other vital protein that would be required in a self-replicating cell. All right. This is the so-called simple cell. Now, when I was first introduced to the cell, it was in 1950. Now, I know I don't look that old, but I was actually alive in 1950. And I was looking through a light microscope, a monoscope. It wasn't even a binocular, it was monocular, and I was looking at cells. And what they looked like was no more complex than a hard-boiled egg. I mean, they had a cell membrane that looked, for all the world, I couldn't call it saran wrap because it hadn't been invented yet. But it looked like what it ought to be saran wrap. It was just an encasement. And then it had a nucleus that would be the yolk of the egg. And then it had this cytoplasm, which was salt and peppered, like as if you added a little salt and pepper to the yolk, or the white of the egg. And that salt and pepper, we didn't know what that stuff was. It stained, you know, blue, and it stained red, but we didn't know what it was or what it did. Well, along came the electron microscope when I was in medical school. And that's what the simple cell really looks like. Now, you'll notice the cell membrane is not the least bit, it looks kind of like this is the progenitor of a porcupine. But it isn't. It's just a cell that could be in any animal or any human on the planet. And the cell membrane is a highly organized and intricate piece of equipment. We used to think it just kept in the things that should be in and kept out the things that should be out. But it functions, allowing things that ought to be in to come in through channels and getting things out through microtubules that spit out the things that oughtn't be in there anymore. Okay? And plus, it has a whole lot of receptors on the surface. Now, we've been talking here about insulin, and so if this were a muscle cell that needed some energy, there'd be a receptor about there, and the insulin would come along and bind to the receptor, and it would open the gate for glucose to enter the cell, where the glucose would go into the mitochondrium here and be broken down from its component of C6H12O6 into carbon dioxide, water, and energy. carbon dioxide would go out into the bloodstream and go to the lung and be exhaled, and the water would go to the kidney and be excreted, and the water would go into the ground and go to the root of a plant, and the carbon dioxide would go into the air and go to the leaf of a plant, and the two of them would get together in the chloroplast, where ultraviolet energy would bring it all together and make more glucose for the whole thing to start all over again. And that's supposed to have happened spontaneously because of a lightning bolt. Well, let's leave the cell membrane and go inside, and we're going to go in here. This is the brain of the cell. This is the nucleus. And that's where we will find the nucleic acid component of DNA. DNA is the storehouse of all information in the cell. DNA makes up genes, and genes make up chromosomes. And in a human cell nucleus, there are 46 chromosomes, 22 pairs of autosomes that determine self, auto is self, and 2 sex chromosomes that determine gender. Now, by the way, in the evolutionary theory, nobody has ever come up with the notion of how gender developed. How did we get XX and XY? And how come we have XO, which produces Turner syndrome, and XXY, which produces Klinefelter syndrome, neither of which are an improvement on the human condition? These are questions that ought to be asked in your biology class, but no one dare ask them because you flunk if you do. All right, now let's talk about insulin. The DNA here in this nucleus knows how to make insulin. It's got the information. It knows how to do everything. And yet it doesn't do anything. You know, we used to talk about 4 categories of physicians. There's the internist like me, who knows everything and does nothing. And then there's the surgeon, who does everything but knows nothing. There's the psychiatrist, who knows nothing and does nothing. And there's the pathologist, who knows everything and does everything 24 hours too late. Well, the nucleus is kind of like an internist. Knows everything, but does nothing. But he doesn't keep the information to himself. Because knowing how to make insulin, RNA, which is circulating out here through microtubules in the cytoplasm, RNA comes in, and messenger RNA comes in and sidles up to the DNA, and the information is transferred from the DNA to the RNA. And I'll show you pictures of those 2 chemicals in a little bit. But anyway, after the messenger RNA gets the information on how to make insulin, it moves out here to, this is called the endoplasmic reticulum. And all those little dots you see that we used to think were pepper are actually ribosomes. And they contain ribosomal RNA. And so the messenger RNA passes the information on to the ribosomal RNA in the endoplasmic reticulum, and the ribosomal RNA now knows how to make insulin, just the way the nucleus knew how to make insulin and passed it on to the messenger RNA how to make insulin, and the messenger RNA passed the information on to the ribosomal RNA. But guess what? The ribosomal RNA can't make insulin because it doesn't have the raw materials. It's kind of like Lordstown out there, northeastern Ohio. The line knows how to make cars, but it needs fenders and bumpers and gear shifts and blah, 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 and to put them all together to make the cruise. So the ribosomal RNA calls upon transfer RNA, kind of like a trucking company. Nice name, transfer RNA. And transfer RNA goes around and gathers the necessary amino acids to bring into the endoplasmic reticulum, where they line them up just right to spit out insulin. Now actually it doesn't spit out insulin, I lied. It spits out proinsulin, which then has to go to the Golgi apparatus, where a piece of it is broken off so it becomes functional insulin. Then, when the body calls upon it, the Golgi apparatus releases the insulin to a microtubule, which goes out and it spits it out into the circulation. Now that all had to happen in that first primordial cell. Remember, it is self-sufficient, self-replicating. It had to have all of the stuff that I've just talked about in order to function. And it all had to happen at once. Now, it gets interesting, if it isn't interesting yet. There's DNA. Now, DNA is a helix, a wound-up helix. And it's composed of these sugar groups that form the backbone of the DNA, and then some nucleotides in here bound together with phosphate groups, and the whole thing, depending on how many genes the creature has, are wound up inside that nucleus. Now, we talked about DNA, which is the double helix here. This is RNA. Well, let's say that this is messenger RNA. It's just come up alongside the DNA to get the information on how to make insulin. And then it goes out and meets another of its kind in the ribosome, and then they call upon transfer RNA to help bring the materials in for the production of insulin. Now, we're getting down to the crux of the matter here. Remember what the simple cell looked like. Remember how much DNA was involved in the simple cell. The DNA is simply a building block of a gene, and gene's the building block of chromosomes, and that's all. Well, let's look at the probability that one rung in that DNA ladder, which has 2 5-carbon sugars, 2 phosphate groups, 2 nucleotides with all of the specific bondings. What is the probability that one rung could have evolved spontaneously? One rung. Now, just to give you an idea, we're talking here about a lot of DNA. The smallest creature on Earth, I'll show you a picture in a little bit. The smallest living creature on planet Earth has over 1,350 genes. Humans have around 25 to 30,000. The Human Genome Project determined that if all of the DNA in one of us was unwound and laid end to end, it would reach from Miami, Florida to Seattle, Washington. That's a lot of DNA. Now we're just looking at one rung here, friends. One rung. What's the probability that one rung could have come about by chance? Well, let's keep it simple. That's the old acronym KISS. Keep it simple, stupid. We're going to keep it simple. Let's suppose that we have 3 blocks here, and each of the blocks has, on all of its 6 sides, one of these 3 letters, A, B, or C. Now, if I were to throw those 3 blocks out on the floor, what are the odds that it would line up A, B, C instead of BCA or BAC or CAB? Well, the mathematical odds are figured this way. 1 times 2 times 3. 1 out of 6 tries will get it right. If you add a 4th block, it's 1 out of 24 tries. You add a 5th block, 1 out of 120. You add a 6th block, 1 out of 700. Now we're looking at about 100 proper sequences in that one rung of DNA, of substances and bonds. What is the likelihood that 100 things could line up spontaneously in order to give us just one bit? By the way, there's that creature. It's got a nice name, Plagibacter ubiquii. He's called ubiquii because he's ubiquitous. He's all over the place, in the water, all over the place. And he's a particular type of an organism. By the way, you notice he doesn't have a nucleus. That means he's, well, I'm not going to get into that right now. We'll talk about that later. But there are 2 different types of cells. He's a prokaryote. See that? And he's got 1,354 genes. That's the smallest possible form of life on planet Earth. And as I say, humans have 25,000. Now, what's the likelihood that 100 things would be lined up properly? Well, I'm going to do the math for you. You don't have to, do like I just did with 1 times 2 times 3. 100 things. Here's the figure. 1 times 10 to the 364th power. That's 1 with 364 zeros behind it. Now that's a pretty big number. Now, let's put that in perspective. You know how many seconds there are in 4.5 billion years? Well, I'll tell you. 1 times 10 to the 18th. And when you divide powers, you subtract. So, how many tries do we need in 4.5 billion years? 1 times 10 to the 346 tries per second for 4.5 billion years to get 100 things aligned just right, and you only have 1 rung on a DNA ladder when you need 1,352 genes that have a lot of rungs and a lot of DNA in order to have life. Now, I've been giving this lecture for over 40 years. You'd think I'd be tired. But you know what? I never cease to be amazed by the questions I get after me. Like one guy who said, well, how do you know it didn't happen on the first try? I don't. The question is to you, and does anybody? And what are the odds? And if you think it happened on the first try, I suggest you go to Las Vegas. All right, now we're going to get into some interesting things here. We're going to talk about preventing congestive heart failure and the intricacy and design and mutual dependence of a bunch of different systems in the human body. This, is, of course, is the human heart. Human heart receives blood from the upper half of the body through this vessel called the superior vena cava, and from the lower half of the body through the inferior vena cava, and they empty into the right atrium. They pass through this valve, called the tricuspid valve, into the right ventricle, and they're pumped into the lungs through the pulmonary artery, where the blood releases the carbon dioxide and receives oxygen. The blood then comes back through the pulmonary veins, 4 of them, into the left atrium, passes through the mitral valve into the left ventricle, and is ejected into the aorta through the aortic valve. Now, that's a normal circulation. Now, under normal circumstances, this goes on quite well. If the muscle becomes diseased through multiple myocardial infarctions or through toxic substances that destroy the muscle, or through infection. We used to, when I was in medical school, there was a thing called rheumatic fever. We don't see rheumatic fever anymore because of penicillin. But we had a lot of people who had diseased left ventricles and valves due to rheumatic fever. Well, many of these diseases can create a situation where the left ventricle becomes weakened and starts to fail. Now, long before you or I would be aware that our ventricle was failing, some intelligent designer put up here in the left atrium pressure sensing cells. When the left ventricle starts to eject less than it receives, that is the start of congestive heart failure. And the pressure rises not just in the left ventricle, but goes up into the left atrium, where these pressure sensing cells send a signal up the nervous connection between the heart and the brain, sends the signal to a place in the brain called the hypothalamus, which then sends a signal to the pituitary gland, the posterior pituitary, which shuts off a hormone called antidiuretic hormone. Now, antidiuretic hormone keeps us from making urine and causes the blood volume to rise. The last thing you want in congestive heart failure is antidiuretic hormone doing its job. What we want to do is shut off antidiuretic hormone so that the kidneys start emptying out the excessive blood volume and a diuresis occurs. And that occurs many times before the patient ever has that first clinically evident episode of congestive heart failure. And that requires the mutual dependence of the cardiovascular system, the nervous system, the endocrine system, and the renal system, all working together to save the patient. This is called the renin-angiotensin-aldosterone system. This is an interplay between liver, lung, kidney, heart, pituitary, and the entire circulatory system of blood vessels. Here's what happens. Blood passing through the kidney keeps our blood pressure controlled by this mechanism. If there's a decrease in renal perfusion, that is a decrease in the amount of blood passing through the kidney, there is a very highly complex, intricate apparatus here called the juxtaglomerular apparatus. It releases a substance called renin into the bloodstream. The liver is constantly making this substance called angiotensinogen. Renin converts angiotensinogen into angiotensin 1, which is totally inert. It does nothing. But the lung produces something called angiotensin converting enzyme, which converts angiotensin 1 to angiotensin 2. And this does a lot of things. First of all, it increases the heart rate, which increases the cardiac output, which increases perfusion of the kidney. It also goes to the kidney and increases the tubular resorption of sodium, which increases water retention. It goes to the adrenal gland and causes the secretion of another hormone called aldosterone. It also goes to the adrenal gland medulla and causes the secretion of epinephrine, which causes arterial vasoconstriction. It goes to the pituitary and causes ADH to be secreted, not turned off, so that there is no diuresis and water is retained through the collecting system of the kidney. And all of this increases the blood pressure, which shuts off the renin. Pretty intricate. And that's just one of the simple functions of the human body. All of this supposedly evolved by chance. My friends, through more than 50 years in the practice of medicine, and through 43 years of being a Christian, I have to agree with the psalmist who said, "We are fearfully and wonderfully made." Thank you for your attention, and I'll take any questions you have.